Hello, it's Dr. Mandy, and today we're going to talk all about gut health. So let's get into it. If this is your first time joining me, be sure to click that subscribe button and like this video if you find this information helpful. Today, I'm going to be interviewing a world-renowned research microbiologist who's been in the nutrition and supplement industry for over 17 years. He's currently involved in over 9 clinical trials on the human microbiome and probiotics. Kiran offers his extensive knowledge on the human microbiome and how it relates to health and well-being. I am truly honored to have him on today, and I truly hope you enjoy this interview. Hey, everybody. It's Dr. Mandy here, and I'm here with Kiran Krishnan with Microbiome Labs. Super excited to have this interview today. I have been a big fan of Microbiome Labs for a long time, as long as I've been in the health world. And today we're going to talk about gut health, of course, but also we're going to tie it into bladder and vaginal health. As you guys know, the gut and the bladder and the vagina are linked. And a lot of the problems with bladder and vaginal are often tied to gut dysfunction. And those of you listening who are my patients know that I rely heavily on spores to help reestablish the microbiome or flora to one that's friendlier. And I actually stole one of your quotes, Kiran, about building the foundation. You said that a long time ago. And you know, what I like about that is I always like to use analogies with my patients about like when you build a house, you're not going to build it on something that's unstable. You want to build it on a stable foundation. And what I find, and with all my patients, when they start with me, I always start with your products, the Mega IgG 2000 and then the HU58. Those are the first two everyone starts even before we get a lab result, because I feel like it helps build the foundation so protocols can work better. So I stole that from you. So anyway, so I'm going to introduce you to Kiran in just a moment. But for those of you who don't know me, let me tell you about my story. 15 years ago, and I can't believe it's been that long, I was diagnosed with interstitial cystitis and bladder pain syndrome. And it's the worst condition I think anyone can get afflicted with. And I'm not trying to minimize other people's conditions, but I'm going to tell you this one is horrible and really disrupts your life. I was told that I would have this forever, and I was given the lecture of like what they can offer me. So it was Almiron and bladder installations, and they wanted to do a cystoscopy, and none of that appealed to me whatsoever. And I did end up taking loads of antibiotics for some UTIs that I was pretty much getting every month. I mean, it was just constant UTIs. It was a very scary time in my life. And I suffered longer than I really should have. But then I discovered functional medicine through Reed Davis's FDN program. So that was one of the first programs I started with. And I learned that my condition was strongly tied to my gut. So that was really eye-opening. Fast forward about 10 years after my diagnosis, and I find out I have significant dysbiosis. I have really bad candida and mold overgrowth, and of course, leaky gut. And all of this contributed to oxalate toxicity. So I do believe that my intolerance to oxalates, because a lot of people in that community, they just think it's all diet. And I'm here to tell you that it's not all coming from the diet. I truly believe that the gut dysfunction is really the underlying problem with this. And so that's how I found out about the oxalates. And I also had a lot of nutritional deficiencies. I had poor eating. I had an eating disorder when I was younger, and then my gut health. I was born C-section, I was bottle fed, so I didn't start off under good circumstances. And, you know, all this inspired me to continue with my education and to obtain the doctorate and then the certified nutrition specialist, which is the flagship certification of the American Nutrition Association. And then I came across a low ox, low histamine diet. ozone therapy, glutathione, blah, blah, blah. We moved out of the moldy house, brand new home. I mean, it all kind of just came together over time. It took me 3 years, Kiran, to get better, 3 long years. But I was getting better over time. It was just, the whole process took about 3 years. And I'm here to say that I am healed. I don't like to use the word cure. I know people throw that word around. I don't like it because, again, analogies. If someone is obese, they lose weight through diet and exercise. They go back to eating junk food and not exercising. Will they gain weight? Yes. So I feel like it kind of falls under that same analogy. If I go back to doing the things I did, my condition will come back, but I am symptom free and I'm living life finally. I am a big fan of Microbiome Labs, and I use their products across the board. So many things, and I love the probiotic shoes. Just want to tell you that.
Same
I
here.
hate taking pills, so thank you for the food. So anyway, I truly believe the gut is a big root cause of IC and bladder pain. Again, so Hg58 mega IgG, we start there, and really just everything kind of falls in together. Again, I'm because of my own healing, but I also see such success with my patients. So welcome. We are really happy to have you today. So quick, let's do an intro on Kiran. So let's dive into this really in-depth background. He is a research microbiologist and has been involved in the dietary supplement and nutrition market for 17 years. And he's come from a strict research background. That's what we really love about you is your science background when you talk about things. We really appreciate that. And so his background is several years with R&D in the field of molecular medicine and microbiology at the University of Iowa. He's also established a clinical research organization where he designed and conducted dozens of human clinical trials in human nutrition. He's a co-founder and partner in New Science Trading LLC, a nutritional technology development research and marketing company, and the chief scientific officer at Physicians Exclusive LLC and Microbiome Labs. He's developed over 50 private label nutritional products for small to large brands in the global market. He's a frequent lecturer on the human microbiome at medical and nutrition conferences and conducts the popular monthly microbiome series webinars through Rebel Health Tribe Group, and is often seen as an expert guest on national radio and satellite radio, and has been a guest speaker on several health summits as a microbiome expert. And we would love to have you at the ANA Summit next year. He's currently involved in 9 novel human clinical trials on probiotics and the human microbiome, and offers his extensive knowledge and practical application of the latest science on the human microbiome as it relates to health and wellness. And that's why we're here today, and I'm honored to have you on. Thank you.
Thank you so much for having me. It's I've had the pleasure of knowing you and working with you in some capacity for the last few years, and it's also wonderful to see your practice and your career and all that grow, and more often than not in our space, the practitioners in our space come from a place of need, right? Where often it's because they were trying to heal their own problems or their family's problems, and so that's what really drew me to this integrative holistic medicine space as a researcher and a scientific person, because to me that is the quintessential healer is somebody that has deep empathy for their patients, right? And the deep empathy comes from your own journeys. So thank you for sharing that journey of yours. I think it's wonderful to hear that, and I think it'll give a lot of people hope that that you can get better, and
oh yeah, I'll never forget the day I stumbled upon this magazine at the chiropractor, and it was a picture of a sick cartoon, and it says, if you can get worse, you can get better. And the concept has always been there. If you cut your finger, does it heal? And so now I use that analogy with people when they're so hopeless and they come to me, and I say, when you cut your finger, does it heal? You might need a little Neosporin, right? Like there's things you might have to put on it, or antibacterial, but it does get better. So you have an immune system. You just haven't been able to harness your immune system to help you with your bladder. And that really gives people hope, and that gave me hope. And so we're really here to talk about that today. And our audience is mostly suffering from bladder pain syndrome, but also we have vaginal issues and gut issues as well. I have used the Biome FX test with many patients, and I am excited to have the opportunity to test the new vaginal test, which I'm actually working on. I got about 6 of them that came in, and I'm interested in looking deeper to see what's going on there. I'm sure we'll find some good trends. So, Microbiome Labs, how did it start?
Yeah, similarly, where there was a significant need, it was really born out of a need in the marketplace. So prior to Microbiome Labs, I owned a research company, New Science Trading, and then a clinical research organization as well called LiveSmart. And we were developing technologies, a lot of them ingredients and ingredient technologies for other companies. Then I was also designing and running clinical trials for nutritional companies as well. We got a call one day from a large multinational company that wanted us to do some development and research work around probiotics. This was probably back in, I'd say, like 2007 or 8. And I was actively working on a couple technologies at the time, vitamin K27, which a lot more people know about now, nattokinase, a number of different therapeutic enzymes. And then this company had said, hey, what we want to do is hire you guys to figure out the probiotic market for us, where they were getting lots of pressure on the shelf, the store shelf. They wanted to understand what is a true probiotic. Is it going to be a refrigerated product? How many billion CFUs? What strains they should use? All of that stuff. So we went about and set out a study on probiotics in the marketplace and started digging into how are these products formulated. Why do companies do it the way they do? Why 50 billion? Why 100 billion, right? Why 15 strains? What's the deal with stuff in the refrigerator? Should it be on the shelf or refrigerator? Soil-based versus this and that, right? So we went through all of that iteration, and then we really came down to this idea that there was a big missing link in probiotic therapy, which was identified that we identified was the spore component to it, right? Nobody was using spore-based strains at that time. There wasn't a single product out except for one called Digestive Advantage, which had just Bacillus coagulans in it. So in this huge array of probiotics out in the marketplace, that many of them looked very similar to one another, just kind of slightly different variations of one another, there was only one product on the market that had one spore in it. And so that was very curious to us, and we started digging into the idea around spores and their use as a probiotic. And we came back to this company and said, hey, after a year and a half or so of work, here's what we think is the next generation of probiotics. Here's all the issues we see with the existing probiotic marketplace. And this is how you formulate a product. This is how you resolve the problem. Because to me, what all of that work said was that we were missing the boat on an important therapeutic component that could really help people, but the vast majority of companies were just doing it completely wrong, right? They were just throwing stuff together, hodgepodging a bunch of different strains. Everyone's fighting for this higher CFU count for no good reason at all, right? Like 100 billion, then 120, then 150, and it just keeps going up and up and up. And so we just saw too much nonsense in that space. Now, fortunately now, when we think about it, but unfortunately at the time, by the time we got back to the company, they had been sold. So the new owners of the company said, this is nice, but we're not going to do anything with it. So that left a big gap. And it was hard for me to go to sleep at night knowing what I now knew about all of the billions of dollars of probiotics being purchased by people, and the fact that vast majority of them were probably not doing anything, and that we're missing this whole therapeutic opportunity with the spores. So we started contacting companies in our space that I knew that I'd worked with in the past, in one capacity or the other, and most of them just could not wrap their head around it, right? They were like, we can't sell a probiotic at 4 billion CFUs. Our probiotic is 30 billion or 50 billion or 100 billion, right? For them, they'd have to sell 4 billion at like $3. They were associating value with the CFU count, and they couldn't wrap their head around what a spore was and how they would educate their marketplace about it. So everybody just kept rejecting the idea, and so that's when I met my business partner, Tom Bain, who's a clinician here in the north side of Chicago. And what was interesting about him, when I met him, he has a gut practice, meaning 90% of what he does is gut related. And yet, as a clinician, he wasn't using a single probiotic in practice. this, right?
Yeah.
And that was so curious to me because I was like, well, how are you not using a probiotic? He's like, nothing works.
Yeah.
I've tried all kinds of stuff, right? So he was using ferments and he was using immunoglobulins and different things, but he was not using a probiotic. So we sat down together and said, hey, we got to bring this to practitioners. This is what we came up with. Here's a formulation. It was the Megaspore formulation. And we decided to run with it and do it. So that's how the company started. And our basic philosophy is, let's solve some big health problems, right? Even in the functional medicine space, we can very quickly get away from root cause medicine, right? It can quickly start to look more allopathic depending on the condition where we're chasing symptomology. We see that in SIBO, for example, right? SIBO is a very good example of people just chasing symptomology and taking a very allopathic look to it. And so, we started saying, okay, how can we bring about an understanding of root cause problems, a lot of which is, stems from the microbiome. So we become a microbiome company to help discover these root cause connections and then educate clinicians on what those connections are, and then hopefully create products to resolve those problems, right? So it's purely born out of that need, and the company will continue to grow and scale only through that need, right?
So upstream medicine.
Yeah, exactly.
Upstream
and
medicine.
everything we do is about finding new solutions for true root cause problems. I don't care how hot an item is in the industry, in the marketplace. We're not going to do it just because it's popular, right? And we already have too many of the companies in our space that just kind of copycat each other, and before you know it, they have 300 different products. For us, it's about finding those therapeutic gaps and filling them with innovation. So yeah, that's really where the company has come from.
That's great. And that's why we love you. So let's define probiotics, prebiotics, psychobiotics, and symbiotics real quickly.
Yeah, of course. So probiotics are defined as a live microorganism when administered at adequate amounts confers a health benefit to the host, right? So there's a couple of components there to unpack. Number one, it has to be a live microorganism, not only alive when you administer it, but then alive when it takes on action in the body. Then the second part of it is adequate when administered at adequate amounts, which means there's a dose dependency effect in there, and then it confers a health benefit to the host. So you have to be able to measure some sort of health benefit to the host, right? So for something to be truly a probiotic, it has to be a live strain, survive through the gastric system, get to the site of action alive in the intestines. There should be a dose dependency on its efficacy, and you should be able to demonstrate what effect it has on the host, and hopefully it's a positive effect, right? So that's a probiotic. Now, if we go by that definition, which is the scientifically accepted definition, I think that was established by the World Health Organization, 90% of the probiotics on the market, 98% don't meet the definition of a probiotic.
Yeah, I don't think they even survive the gut, right?
They don't survive the gut. No, that was one of the first discoveries we made when we were doing our research on probiotics. We took 40 of the top selling probiotics in the marketplace. We put them all through gastric survival testing, which is a standard mechanism of testing under US pharmacopoeia. And then 98% of them all died in the stomach, right? So they're not even getting to the site of action alive. So right off the bat, we're like, okay, these don't actually qualify as probiotics to begin with, right?
Oh, okay.
Yeah, they're not engrafting. They're not necessarily conferring a health benefit to the host. And if there's a finished probiotic product out there, a formulation that the company who's selling it hasn't demonstrated that finished formula has a health benefit, then it, again, does not meet the standard, right?
It does not.
What a lot of companies do is they'll just go, hey, this strain has some research on it, that strain has research on it, and this strain may or may not, and then they'll throw a couple of strains that have some research on it with a few other strains to bump up the CFU count, and then they will just assume that has positive clinical effect.
Ugh.
You can't.
That's frightening.
Yeah, it's a huge leap. It's a huge scientific leap to do that, right? So that's probiotic. Prebiotics are foods for the good bacteria in your gut. They're fibers, soluble, insoluble, oligosaccharides, really, really important components.
Did you hear that, everybody? Fiber.
Fiber.
Okay.
Good old fiber.
I'm always preaching fiber, so that's why I had to.
Absolutely. Well, it's amazing how much of a health impact adequate fiber can make in
Mm-hmm.
an individual, right? It completely changes the game of what's happening in your gut. And here's the reason why. This is the simple explanation for it. And most people think of fiber as just kind of this roughage that cleans out your system and moves through. What they fail to remember is that fiber is the starting fuel for a huge array of necessary compounds to function as humans, right? So more than 50% of the compounds that we need to function as a human. So in your blood right now, if you pulled out your blood and you looked at all the compounds in your blood that your cells are using and all the components of your body are using, 50% of them were synthesized in your gut. You
Mm-hmm.
don't get them from diet, right? You don't make them yourself. They were synthesized by bacteria in your gut. And those bacteria require fiber in order to make those compounds, right? So a good example of that is butyrate, propionate, acetate, so your short chain fatty acids,
Yep.
right? You can't live without adequate short chain fatty acids. And if you're not making enough, there's all these diseases that go along with not having adequate amounts of short chain fatty acids. Butyrate, for example, everything under metabolic syndrome is dramatically increased in terms of risk when you have inadequate levels of butyrate, right? Butyrate controls your metabolism, your fat burning, your satiety, your ghrelin and leptin functions.
Oh, fuel for your epithelial cells.
Everything, yeah. Fuel for the primary fuel for the lining cells in your gut. The primary fuel for reproducing your mucin layer, which is a really important structure, which I'm sure we'll get to at some point. And it's a primary fuel for the cells, your dendritic cells and macrophages that float around your body trying to protect your system, right? They use butyrate from the gut as their primary fuel.
Wow.
Right? So many things are tied to butyrate, and you cannot make it yourself. You need bacteria in the gut to make it for you. So, you cannot discount fiber, because if you do, you are not putting in adequate amounts of the starting material that your bacteria needs in order to produce all these compounds, right? So that's prebiotics. And then symbiotic was the other question. So
symbiotic. Yep.
Right, symbiotic. So a true symbiotic is a probiotic and a prebiotic mixed together.
Oh.
And so what you have to
The
chews, exactly. So the chews are
I love the chews. I have to be
example
honest.
of that. I love them too. I mean, they're so easy.
Is there a limit to how many you can eat?
Absolutely not. No.
Okay. Because I've been eating like 2 or 3. I'm like, gosh, I hope this is okay.
I do on average probably 4 a day of this.
Okay. All right. I feel better.
I love them. So the chews have K27 in it as well. Very
nice.
It's got great level of prebiotic in it, and of course the probiotic. Now, for it to qualify as a symbiotic, what you have to demonstrate is that the prebiotic in the product has to enhance the function of the probiotic, right? So you demonstrate what the probiotic can do on its own, and then you demonstrate that when you add in the prebiotic, that it enhances the function.
Oh, puts fuel on it.
Yeah, exactly. So it has to be an additive effect.
Yeah.
Right. If the prebiotic in it just helps other bacteria and doesn't enhance your probiotic, then it's just a combination prebiotic probiotic. It's not a symbiotic, right? So symbiotic has a scientific definition to it. Now, we've demonstrated that the chews truly are a symbiotic, because what we've shown is that the probiotic has all these beneficial functions, the megaspore. We've shown that it increases short chain fatty acids like butyrate, propionate, acetate, increases diversity, and all these other things. Then we've also published a study showing that when you add the prebiotic in, it more than doubles all of those effects. So it's enhancing the functionality of the probiotic, which is a critical component. And I think the last one you said, was it postbiotics?
It was actually psycho.
Oh, psychobiotics. Okay, and then we should talk postbiotics.
Yeah, let's do that too. Yeah.
That's another one. Yeah. So psychobiotics are unique probiotic bacteria that have a direct impact on the brain. And we have a psychobiotic called ZenBiome. It's one of the well-studied psychobiotics on the market. I think we have about 6 published studies on this particular product. What you have to be able to demonstrate in this is that there's a alteration or improvement in brain chemistry and hormones related to brain function. And then we go one step further, where we can demonstrate that the probiotic actually changes brainwave activity. So it actually puts your brain in similar to what the kind of calm state that you would get from meditating, for 40 minutes or an
Oh,
hour.
everybody needs that right now.
Everybody needs it, right? The whole benefit of meditating, or at least the biggest benefit of meditating, is it puts your brain into more of a theta wave band,
Yeah.
right? That's that flow state where you're very calm and your coping centers of the brain are really active, and artists and all that call it the flow state, where they're not really having to think about stuff, but they're working at a very high level. And this probiotic actually puts your brain in that state.
Got
So
it.
it becomes easier for you to get through the day and deal with all the different stresses and pressures of the day. Things roll off your back much easier. And of course, it reduces all of the inflammation associated with stress response and HPA activation.
I think I have to add Zen Biome to my protocols more often.
Oh, it's the
Yeah.
best. It reduces neuroinflammation, and so many people now have significant neuroinflammation, especially when we start talking about COVID long haulers and all that. And lots of people have tryptophan metabolism issues where tryptophan is being metabolized to quinolinic acid and renin, right?
Through under inflammation, correct?
Exactly. That's it.
Yeah.
Flips that switch. Zen Biome.
COVID has upregulated that.
Absolutely. That's in fact the core mechanism behind COVID long haulers and why people have continuous neurological symptomology. It's because they've shifted tryptophan metabolism. Now they're taking tryptophan and making it a neurotoxic product rather than melatonin and serotonin, right? Which is protective. And so,
quinolinic acid, not to interrupt, but it can increase pain sensitivity.
Yeah.
For people who have the bladder pain and elevated quinolinic acid, it's heightened. So it's much worse than it might actually be.
Totally. Yeah. Because not only are you getting the pain signals, but you've got neuroinflammation baked in with it, right? Which makes everything much worse. So the Zen Biome can be extremely helpful. We also know that chronic pain causes anxiety and depression, right? And it leads to that
vicious cycle.
Exactly. It's a vicious cycle. And so this product reduces anxiety, reduces anxious response in people's brains. So it can be a really, really beneficial neurocalming psychobiotic. The last one, postbiotic that we talked about. So postbiotics are actually earlier on were defined more as the byproducts that bacteria produced in the gut, right? So short chain fatty acids, like we talked about, and vitamins and hormones and other things that microbes makes. Now, about a year and a half ago, there was a change in the definition where dead bacteria that have a effect in the body are also called postbiotics.
Okay.
So the ones that aren't alive, but they've demonstrated that even in the dead format, they have some functionality. And there are some of those that have good studies on them showing that even in the dead version, they have the effect. We launched a product like that called PyloGuard, which is
I have that. Okay.
It's a Lactobacillus reuteri. What's unique about it is it binds and removes Helicobacter pylori from the stomach. But it's not even alive. It's dead. It's fully intact. The cell is intact. The membrane structures and all are intact, but the bacteria is actually dead. So there's no sensitivities to stomach acid and temperature and storage and all that stuff. You take it, it actually goes in, and it as it comes across H. pylori, it binds and aggregates to it.
Oh, it's like a binder.
Yeah, but it's only specific to H. pylori, which is what's so interesting about it. So that one we have, I think, 8 published studies on that particular product. So really, really effective. And we know that H. pylori is a big issue as a dysbiosis driver in percentage of people, right? At least 50% of adults.
I had that when I was a senior in high school.
Yeah. And, a lot of times it's because most people get infected with it sometime around 7 or 8 years old at home normally, right? It's very transmissible in people's homes and schools and things like that. It's transmitted through saliva contact, so sharing utensils and spoons, and then also through the fecal oral route. And there's lots of fecal contamination all over the place. We are accidentally coming across people's poop and consuming it.
Lovely.
Touching doorknobs and things like that, right? Someone goes to the bathroom, doesn't wash their hand, and they
watch the little cartoon on YouTube, the sneeze. You'll wash your hands forever.
That's what I do. I have to wash my hands. And it bothers me. I go all the time. If you go to a public restroom, you see people coming out of the stall, knowing what they were doing in the stall, walk right out of the bathroom. Right, they don't stop and wash their hand. It's horrific to see that. But I'm all for sharing microbes, but not in that way.
Not pathogens.
Not pathogens, exactly. So that's a postbiotic. And so now, certain types of dead bacteria can have an effect. It's rare. It's not, most dead bacteria probably don't have much of an effect, but there are some strains that have been well demonstrated to do that.
So spores are alive, correct?
They are, yeah.
Okay.
They're alive in their own unique way. So when you find spores whose natural home is the gut microbiome, so you can have spores in many different environments, right? You've got spores that have adapted to being in the highest peaks of the mountains. You've got spores in the Tibetan plateaus in the North and South Pole. They're basically ubiquitous all over Earth. In fact, there's some evidence that they might have been the earliest forms of cellular life on Earth.
Oh.
Because you can find components of these types of bacteria on meteorites and things that crash
Oh.
Earth, right?
Wow.
And there was a study done where they were looking to see if Bacillus subtilis could survive interstellar travel in space, in the vacuum and cold of space, and they could survive up to 7 years in space.
How do they do that?
They just go into the spore state, right? This is what's amazing about them.
Yeah.
They're not in their ideal growth environment. So in our case, the spores we work with, their growth environment's the gut. The moment they come out of the gut, so like through defecation, for example, they sense that they're no longer in the gut, they go into the spore state.
Dormant.
Means they're metabolically inactive, right? They wrap themselves with the protein calcified coating. So they have this hard, really hard to penetrate coating, and then they just sit in that dormant state until they get back in the gut. They could sit like that for hundreds of millions of years.
So you could put your probiotic outside on the counter for 100 years.
100 million years.
100 million years.
Yeah.
But no refrigeration necessary then.
Exactly, right.
Yeah.
And so the oldest spores, there were two really cool studies on some of the oldest spores found. The oldest ones were found in a cave in Southern California. Scientists have been going into deep areas of caves looking for new bacteria. The reason they're looking for new bacteria is they're trying to look for new antibiotic compounds, right? Because so many antibiotics have now developed resistance.
Yes.
And microbes, pathogens have developed resistance because they overuse.
We're in a mess.
We're in a mess there, right? And we are in a danger of going to what they call a post-antibiotic age, where there will be no longer antibiotics in the market that are effective against certain pathogens.
Wow.
That's super scary, right?
Yeah,
because in the pre-antibiotic age, which is prior to penicillin, you could die from a simple sinus infection. You could die from a cut on your hand, this just simplest things. Now we are in danger of going there because of our abuse and overuse of antibiotics. So most antibiotics, 99.9% of antibiotics and their compounds and how they work come from microbes, right? So microbes, especially bacteria, produce a lot of antibiotics. That's how they compete with each other in their little microbial environments. So we are trying to learn about new antibiotic type compounds from trying to study bacteria. And so they go into these deep recesses that humans have never touched, and they're taking samples of bacteria from the cave walls and other things, and they're trying to synthesize, they're trying to inoculate and study these microbes to see if they produce some unique antibiotic compounds so we can learn about new generations of antibiotics. One of the studies that they did, they had these salt crystals in the caves that they extracted from the caves, and then they melted down the salt crystals, and there was microbes in there, spore-based bacteria, and they could still plate them. They were still alive. Those were dated to be 500 million years old.
Wow.
So they were in a dormant state in the cave for 500 million years. And then now that you plate them and you put them in the right condition for growth, boom, they come back alive again after that amount of time, right? It's mind-boggling when you think about it. Another cool one, it's actually kind of reminiscent of the movie Jurassic Park. In South America, they were doing these studies on the gut microbiota of ancient insects, because there are these whole honeybees and other insects that were completely fossilized in amber, right? So they could drill through and drill into the gut of these insects and pull out the types of microbes and things that live there. And so they were able to drill through this fossilized amber, and inside is a whole honeybee, and they drilled through, they pulled out samples from the gut of this honeybee, and sure enough, they were able to plate it, and those microbes were still alive, and the honeybee had Bacillus spore-based probiotics in its gut.
Very interesting.
Right? And that was 50 million years old. So it had been fossilized for
Yeah.
50 million years and still alive now, right?
Huh.
So these spores have been around forever.
Yeah. Yeah.
They
Our DNA are programmed to know what to do with this.
100%, which is
Yeah.
so interesting, right? Because we have a very limited number of genes for the kind of species that we are. We only have about 22,000 functional genes, right? And it might sound like a lot if you're not familiar with genetics, but keep in mind that an earthworm has somewhere around 25 to 30,000 genes, right? So we have fewer genes than an earthworm does.
Yeah.
Which then begs the question: how are we so sophisticated? How do we do all the things we do? Well, we have about 2.5 million microbial genes in our system, right? So most of our capabilities are coded for by bacteria, viruses, fungi, and so on that exist in our system. Mostly bacteria, but there are other microorganisms as well. But among that small array of genes that we have, we do code for receptors in our gut lining that welcome in these spores, because we've had this long-term symbiogenic relationship with them, right? So I've used that term before, symbiogenesis.
Might as well define it while we're here. Yeah, what is it?
Yeah, absolutely. So it's
we know what symbiosis is, but maybe some people don't. Yeah.
Yeah, so it's a combination of the two words, symbiosis and genesis. So symbiosis being this mutualistic relationship between two organisms that have been forced to live together. And then the genesis part is the development of that relationship, right? So symbiogenesis simply is defined as a symbiotic relationship that's been developed by a forced relationship between the two organisms. So we as a species evolved.
Could that be like us and canines?
Us and what? Sorry?
Canines, dogs?
Yeah, absolutely. Now, even it's more kind of mutualistic in that there's a benefit to both, but it's not necessary, right?
Okay.
Like we don't need a dog to survive. The dogs don't necessarily need us to survive. However, in the case of microbes, we do need each other to survive, right? So we provide the home for the microbes by consuming them from the environment, and then they basically take care of the home. And that's in large part what the spores do, right? So they've learned over millions of years of symbiogenesis with the human system what the human gut should look like when it's healthy from a microbial standpoint.
Wow.
Right? So they know better what our gut is supposed to look like than we do. And it's uncanny because when we put spores into your gut, and we do, for example, before and after microbiome analysis, you start to see all the kinds of changes you wanted to see in the gut occurring because of the presence of the spores, right? They know if your gut has low diversity, they'll start increasing diversity. They know if you have low levels of keystone species, they'll start increasing keystone species.
Wow.
Right? They know these things. There's a evolutionary intelligence there that we can't engineer, and it's there just because of millions of years of symbiogenesis. It's beneficial for them that our gut is healthy and not, riddled with pathogens and so on, because then the host will be sick, which is their home, right? So it's a really, really cool relationship. Now, the way they do this, and this is really cool, is they do something called quorum sensing, right? These microbes are experts at going into space and reading the microbial environment. They can understand what and how much of other bacteria are present in that area, right? So they actually read the chemical signatures of all the different bacteria. They can understand who's there and in what amount. Now, they do this so well that in the prescription world, since 1952, they've been used as a therapeutic way of treating gut infections, because they have the capability of going in and identifying the one, maybe
spores?
For spores, yeah.
Since 1952, they've been used as a treatment for gut infections?
They have, yeah. The first probiotic spore that was launched is still on the market today. It was launched by Enterogermina, which is a big pharmaceutical company out of France and Germany. They launched a probiotic spore for the treatment of gut dysentery.
Wow.
And it's been in the market for 65, 70 years. They still use it in Europe and Latin America and Southeast Asia. Oftentimes, if you have a gut infection, a doctor there will give you this over an antibiotic because it's more effective, because it doesn't harm anything else.
Right.
Spores know how to look for the pathogens, right? I'll give you an example with our C. diff study. I think that's one of the things that we wanted to make sure we covered, right?
Yeah, for sure. C. diff.
Yeah. So we know Clostridium difficile is a inhabitant in many people's guts. A lot of times it can hang out there and not really cause problems, but the moment you shake up the microbiome, whether it's through a course of antibiotics or other behaviors,
Food poisoning.
Food poisoning is a great example, right? So anytime you mess up the ecosystem a little bit, that is the opportunity that Clostridium will take to start expressing their toxins. And they produce two key toxins. And basically what they do is they create diarrhea. And now the reason they create diarrhea is it gives them an opportunity to clear out space, right? Other microbes fall off as you're having diarrhea. And then they're also trying to eat away at the mucus layer because they need blood. They need the iron in blood to increase their metabolism. So that's why you get bloody diarrhea with Clostridium difficile infection. And it's a horrible thing to get. Lots of people that get it once will keep getting it. There's high degree of recurrence, right? And lots of people die from it every year.
Yeah, I was going to say it can be fatal.
It can be very fatal, and it's hard to treat. Right now, the main treatment is of course antibiotics. They use vancomycin. There may be another antibiotic that they use from time to time, but the recurrence rate is pretty high. Now, fecal transplants have also been shown to be quite effective in treating it. But again, that's not easy to do. You have to do that in specialized places. And there's always
I think there's a lot of risks to the fecal transplant too, no?
That's the big problem, right?
Yeah.
It depends on the donor a lot. You don't know what else.
And are donors healthy? I mean,
right, exactly.
Where are you getting it from? Americans? I don't know.
Right.
I don't know how I feel about that.
There's not a lot of Americans you'd want to get it from.
Right.
Get stool from. And there was actually a famous story on this for a woman that had been suffering, I think, from I think it was colitis for years. She was an athlete, an avid athlete, extremely fit all her life and all that. And she had this colitis issue. So she had all these significant bowel problems. And then finally they did a fecal transplant on her to try to improve the colitis because they had tried all the biologics and the other pharmaceutical approaches. And it helped her colitis significantly. The only problem is she started gaining weight.
I heard about that.
Yeah, and she's never struggled with weight, right?
Yeah.
She's doing her same diet, same exercise.
Wasn't it a family member too who transferred that she got it from?
It was not an immediate family. I think it was a distant family member.
Yeah.
And basically what they realized is that the donor, even though it was quote unquote healthy normal, meaning they didn't have gut issues, didn't have colitis, was overweight and was struggling with weight. So now they transferred the metabolic dysfunction. Right. So yeah, we had.
And she gained weight and couldn't get it off. I remember that story. That was one of the reasons I was like, no, not for me.
Yeah, you just never know. I mean, it's talking about trillions of cells and millions of genes you're transferring in one shot, right? I mean, if that was the very last thing to do before I kicked the bucket and died from something, fine, I might try it. But fortunately, there's so many other things you can do before you ever get to that point, right? But going back to Clostridia, what was so interesting, we've published now 2 studies with Cleveland Clinic. They did the study for us on Clostridia. What they showed is that when you use the Megaspore, our flagship probiotic, in a Clostridia difficile infection system, what tends to happen is the spores go in, they identify where the Clostridia is, so they physically know where that bacteria is. They'll go and sit and actually surround the Clostridia, like kind of wagons around it, right? And then because the Clostridia is also a spore former and is more resistant to antimicrobials and all, which is how the spores would normally kill off some pathogens, what those spores have figured out to do is they start spitting out a chelating agent around the Clostridia to steal minerals away from Clostridia, especially iron. Remember, I mentioned earlier that Clostridia needs iron metabolism, right?
Yeah.
These spores are so intelligent, they know that, oh, here's the pathogen. Not only do they know where the pathogen is, they can surround the pathogen, and they know what kind of pathogen it is because they use a different tool to control that pathogen, right? So we were able to show, or Cleveland Clinic was able to show, a significant reduction in Clostridia growth when you apply these probiotic spores to it, and nothing else gets harmed.
Interesting.
Right? It's such a beautiful natural system that we just have to be smart enough to identify that it happens, test it, and utilize it we can't engineer bacteria to do that, right? So to try to engineer a microbe to be able to find Clostridia, use the right biological weapon against Clostridia and bring down Clostridia and not touch anything else, that's a mind-boggling discovery.
Yeah.
Just we just have to be smart enough to know that nature can do it. We
Yeah.
just have to let nature do it.
Yeah, I don't like messing with nature too much, to be honest with you. So what about Saccharomyces boulardii? I thought that was supposed to be really good for Clostridia too.
It can, yes. So Boulardii does a good job against Clostridia because of its acidification of the gut, right? So it brings down the pH. Clostridia doesn't do as well in a low pH environment, so it slows down Clostridia's growth. It's a great one to use, especially if you're going to have to use an antibiotic. And that's one of the reasons we use Boulardii in our product called Restore Flora.
Love that one. Yeah.
Right? And that was developed actually for nursing homes initially. We had a big nursing home group that said, hey, we have all these residents that, they go to the hospital from time to time from the nursing home for procedure or whatever the issue is. And then such a high percentage of them come back to the nursing home with Clostridia infection.
Oh, interesting.
And so they said, and then they're constantly on antibiotics. And so they said, can you develop a probiotic for us that can be taken with antibiotics and has some functionality against the, number one, the disruption that the antibiotic creates to the gut. Number two is if there is an opportunistic overgrowth like a Clostridia. And so that's why we developed Restore Flora for that nursing home purpose, So if anyone's using antibiotics, it's a great one to use with the antibiotic.
2 hours away from the antibiotic.
2 hours is ideal, yeah. But if they forget, they can definitely take it with the antibiotic. We've done studies on it with the antibiotic itself, or at least
you said it's antibiotic resistance, right? The resistant, you said the spores.
Yeah, what it does. So they're not resistant in the traditional sense that certain pathogens are resistant, right? Where the pathogens have a gene that codes for certain proteins that provides resistance.
Right.
That's not how the spores do it. What the spores do is because they have sensors outside of the spore coat, when they're in the gut, if there is an antibiotic present in the environment, they just stay in the spore state.
Oh, interesting. Okay.
Yeah.
That's good information.
They come out,
Yeah.
So what's good about that, and this is why that is so important, because this is a natural way in which spores function, which means that it's not a gene mutation that provides them antibiotic resistance. If it was a gene mutation that provides them antibiotic resistance, then there's a risk they can accidentally transfer that gene to problematic bacteria.
Right.
Which is what happens when you develop antibiotic resistance to begin with, whether it's in your own gut or in the food system, in the cows and all that we feed antibiotics constantly.
Yeah.
All these microbes start developing antibiotic resistance because they transfer these genes to one another. And the spore cannot transfer those genes. That's part of its core genome, which is how it just normal functions. That's its life cycle.
So I'm going to dive into diversity. So a few years ago, when I first got into gut health, I read this book by Martin Blaser called Missing Microbes. And it was very eye-opening how the lack of diversity and missing the keystone species is negatively impacting our health. And there's a movie that I'm also a biology instructor at the college. I teach as an adjunct, and I have to infiltrate my belief system sometimes with the microbiome when we get into diversity modules, because I think people need to understand the importance of their lifestyle. And there's a movie called Let Them Eat Dirt. You've seen that? I highly recommend it. It is one that's very easy to understand. Children can understand it, as I play it for the college students. I even make it a lesson plan. Talks about some of the stuff that Martin Blaser talks about. So important to play in dirt. Kids do not do that anymore. They're in front of the computer. They're indoors. We're sterilizing everything. Hand wash, what is it? Antibacterial hand soaps. So on the one hand, wash your hands when you come out of the bathroom.
Right.
On the other hand, you don't need to wash your hands after playing in the dirt. But the last piece is Deanna Minich with the ANA. She did a wonderful talk at the ANA conference today about dietary diversity and eating the rainbow diet and diversifying the microbiome. There's another practitioner, Karazian, I think his name is. Karazian, yes, I always mispronounce his name. Wonderful program called the Microbiome Mashup. Have you heard of that?
I have not. No.
It's really cool. I've been doing it with my dogs because my dogs tend to have dysbiosis, all that glyphosate
Yeah.
outside that gives them the diarrhea. So you take about 30 to 40 different vegetables, if you can find them in the grocery store, the stems, the leaves, all the stuff you don't normally eat when you're eating food, and throw them in a blender and make these little, I put them in ice cube trays and throw them in the freezer, and then just throw them in your food, like just little tiny little cubes. And you throw them in a shake, throw them in coffee, whatever. You don't even notice that they're there. So the whole idea is diversifying the microbiome. That's the whole idea with these ideas. Why do we need to do that? Yeah, I mean, one last thing I want to also mention, I was looking to tell you, I thought it was interesting that the Hasda tribe, Hasda tribe eats about 600 different species of plants and animals and 100 grams of fiber.
Yeah.
Wow.
It's crazy.
What is happening to us?
Very similar to Papua New Guinea tribes too, and there's something really scary happening to us, right? And you mentioned Marty Blazer. I love his book, Missing Microbe.
Yeah.
He has a follow-up film.
Oops.
I think it's called. And it's have you heard of that one, The Invisible Extinction?
No.
It's basically a documentary that is kind of the follow-up to the book, but it's more recent information.
Oh, I need to write that down. And The Invisible Extinction?
I believe that's what it's called. Yep.
Okay.
And Marty's part of the developers of it. I saw like a 15-minute version of it, and it's amazing. It's so good. And so let's unpack this a little bit, right? So I would easily say that when you think about the gut microbiome, the most important feature of the gut microbiome, hands down, is diversity, right? That has been shown to be true throughout the last 10, 15 years of microbiome research. Diversity is so important that you can actually predict how long somebody's going to live based on the diversity of the gut microbiome
Oh, wow.
ages, right? There's so much good data now on longevity and diversity. And so you can look at somebody in their 50s and look at their diversity and predict how long they're going to live based on that. And all of the disease risk comes along with diversity issues. And so let's unpack why that is. So, I mentioned earlier that we have very few genetics, right? We have very few genes. We have 22,000 functional genes. That is wholly inadequate to survive as a human. So the vast majority of our genetic capabilities, our protein making capabilities, our hormone making capability, all of these things, the vast majority of it comes from microbes. And the biggest treasure trove of microbial genetics sits in our gut, right? And if we lose microbes, or we have low non-functioning microbes, we lose a lot of that capability, right? So we lose a lot of genes. It's no different than just taking out your genes and your chromosome and snipping part of it off and putting it back in, right? Whoa, what did we just lose, right? What can we now not do anymore? And so that's a big part of it. The other part of it is that a diverse ecosystem, and keep in mind that the gut is truly an ecosystem, right? And it's like any other ecosystem in the world. The more diverse the ecosystem is, the more stable it is, and the more resistant it is to dysfunction. The more stable and diverse your garden is, the harder it is for a weed to take over and start killing all the plants, right? No different in the gut. When you have a high diversity of microbes in your gut, it's harder for new microbes to come in, or opportunistic microbes to overgrow and take over and create dysfunction in the system. And so even if you just look at it from those two perspectives, number one is the microbiome houses the largest components of our capability. And when I talk about capability, I mean just being human in this space, right? To deal with all the things we're exposed to on a daily basis, to deal with the foods that we consume, to deal with the stresses of life. All of those things require a certain degree of resilience in the body, and most of that resilience is conferred to us from microbes, right? So we can unpack very specific examples. You mentioned oxalate, right?
Right.
So in your gut, if you have a healthy gut, you can eat all the oxalate-based foods you want, because there are microbes in your gut called Oxalobacter formogenes, for example, and other species that actually break down oxalates and use them in useful ways, right?
Yeah.
But if you're somebody that doesn't have that diversity, doesn't have enough of those microbes, oxalates can kill you.
Yeah.
Right? It's that
really, it's a toxin. Yeah, absolutely.
Absolutely, right? So you can go from, You can be somebody that has no issue at all with it. You can eat as much oxalate foods as you want, deal with it, all the way to the other end of the spectrum where it could kill you.
I mean, look at like animals, like horses. That's all they eat is oxalates.
Absolutely. I
You
mean, our
know?
environment and food system has been full of those things, right? You can't imagine our ancestors going, "Oh, I can't eat that. Oh, I can't eat that. Oh, I can't eat that." Right?
Yeah.
Like they're on elimination diets and eat only 6 things. We would have never survived as a species.
Well, I have my opinions about that. They were also probably not eating constantly. They were also fasting, but that's a whole nother
Exactly.
topic.
And again, but here's the thing: it ties in exactly to our conversation. Fasting, in fact, increases diversity in the gut microbiome. And that can be counterintuitive, right? Because you're like, wait, not feeding the gut also increases diversity? It absolutely does. And the reason for that is the microbiome is made up of different layers of fermenters, right? So we've got our primary digesters and fermenters, then we have secondary layer, tertiary layer, and so on. So what happens is when the big bulk macromolecules from food come in, the primary fermenters or digesters are breaking them down, right? They have the enzymes and the capability to break down the big molecules. And then they break them down, and the byproducts of that breakdown feed the next layer of microbes. And then they break those down, and then they create byproducts that feeds the next layer. All the while in this chain, we're producing all of these really important compounds, right? That we need for survival, for functionality, for resilience, for adaptation to the environment. So you have to allow each of those layers of microbes to have their time to do their metabolic function. If you keep eating and you have no periods of fasting in your day, then you're only feeding the primary fermenters. They're the ones that are the most active. The other layers aren't necessarily getting the time that they need to do what they do. And then you're missing whole array of molecules because you don't get all the byproducts from these other sections of bacteria, right? So it's a really important thing to keep in mind is that, number one, you need to feed your microbiome. You're eating not just for your own pleasure and the things that you've tied your mind to as being pleasurable, right? Like, oh, I only like this kind of food, or I
Right.
don't like
I
vegetables,
get that so much
right?
from people. Yeah.
Totally. And I'm like,
Very, very limited diet. I call it homogenous diets, just one
totally.
or two foods, three foods that they're just eating over and over and over and over.
And over again. And it's either A, because they feel like they tolerate those the best, or B, because that's what they desire the most, that's what tastes the best for them, or whatever the texture. But at the end of the day, what we cannot forget is we're not just eating for us and our pleasure, we're eating to feed this massive nutrient factory in our gut. Because remember, more than half of all the nutrients you need to survive are made by bacteria in the gut. You
Right.
cannot get them from food. You cannot make them yourself. You have to feed the bacteria the right substrates, and then they make it for you, right? So going back to diversity in the Hadza tribe that you mentioned, they absolutely eat around 600 different foods annually. So did our ancestors. Most of our ancestors, most of the people we evolved from, ate that kind of diet annually, and they ate seasonally as well, right? Because they basically ate what was available at that time, and they hunted, they foraged, they gathered, they did all of that stuff, right? They didn't have the luxury of being picky. They didn't have the luxury of going, I'm going to be carnivore this month, and they didn't have the luxury of going, I'm just going to be keto for a little while, right? It was all about like survival.
Yeah.
And our bodies are designed with that omnivorous type of survival capabilities. We're supposed to eat lots of different things.
Thank you.
Here's a very critical.
I'm so glad you said that. Omnivores.
Omnivores. It's so important, right? That's how we moved up the food chain. That's how we moved up the evolutionary ladder. If you think about a lion, my favorite guy back here, I'm a Leo, so I have a lion back there.
Yeah.
As powerful and amazing as a lion is as a creature, a lion is an obligate carnivore.
Yeah.
A lion cannot eat plants, right? So if there's a drought and there's no wildebeest and there's no protein, the lion will die, right? And the same thing with the wildebeest. If there's a drought and there's no plants, the wildebeest can't start hunting squirrels and things like that. It's going to die.
Yeah.
But a human in that same situation can dig and eat worms, can grab shrubs and berries and eat almost anything in that area, right? That's why we move up the evolutionary ladder. Now, here's another really important point about that. Part of human evolution, and a very important stage in human evolution, was an incorporation of a large fermentive base in our colon. right? And that incorporation of a large fermentive base, which we now call the microbiome or part of the microbiome, came from bringing in microbes from the environment and allowing them to stay there, right? So our immune system developed this mutualistic relationship, this symbiogenic relationship, where our immune system over time learned that, hey, these microbes can actually be useful. Let's not attack them. Those microbes said, hey, immune system, we're going to play nice. We're going to make all these compounds that are good for the host. So don't attack us. We're going to have this great mutualistic relationship. We incorporated lots of those organisms, and the vast majority of organisms need plant-based material, right? That's how they came about.
I struggle with my field with what you just said, and I'm going to get chastised for saying this, but there's just way too many people who are jumping on this carnivore bandwagon right now, and even the people in the oxalate community, and I can't stand by that. After I started looking at your stool test and I started seeing the saccharolytic fermentation marker versus the proteolytic fermentation, and I saw the keystone species, I saw the lack of diversity on all these people. And then I looked at all the nutrients that the bacteria was not producing, no butyrate at all, imbalanced short chain fatty acids, so low butyrate but high acetate. You want it balanced, right? And so when I started noticing that, and I've never been a big fan of extreme dieting anyway, I believe in balance. That's always been my motto. But again, people are like, oh, carnivore diet. And for the longest time, it's been vegan.
Right.
But you just mentioned the plants, and that's that saccharolytic fermentation, correct?
100%. Yeah. That is taking plant-based material and fermenting it and converting it to things like short chain fatty acids, which are absolutely critical for our survival. Even things like, for example, urolithins, right? Urolithins are a very important class of compounds, especially urolithin A. We'll take that as an example. Urolithin A plays a very, very critical role in the health of our mitochondria, right? Our mitochondria are the powerhouses of our cells, right? And a very clear definition of disease and aging is dysfunctional mitochondria.
Correct.
Right? Yeah. The moment the mitochondria start to die or become dysfunctional, the cell starts to die or becomes dysfunctional, and then the organ that the cell makes up dies and starts becoming dysfunctional, right?
Correct. Yep. It all boils down to energy.
It does, yeah. And the capability of the cell to function, replicate, make nutrients and metabolize nutrients and so on. All of that is dependent on the engines of the cell, right? No different than in a car. A car is absolutely useless if the engine doesn't work, right? It can sit there in your driveway, look pretty, look nice. You can sit in it, but you can't go anywhere. That's how your cells work. Your cells have engines for that reason. The mitochondria, in order for them to turn over, and what happens during mitochondria, they undergo a process of cellular respiration. That is the taking in of glucose and sugar, fructose and so on, and converting it to energy, right? ATP. Basic cellular function, right? Yeah. Cellular biology 101. If your cell can't do that, then the cell is useless and it's dead. The only cells in your body that don't have mitochondria are your red blood cells, because the red blood cells have a singular function that is housing hemoglobin that grabs oxygen and transports oxygen throughout your body, right? Outside of your red blood cell, every other cell in your body has to have mitochondria and have to create ATP. If they don't create ATP, then you're basically dead. And so the mitochondria are the most important, arguably most important component of your cells. But what happens with mitochondria, like an engine of a car, when it's going through a metabolic process to create energy, it produces an exhaust, right? Just like a car's engine produces an exhaust that can be noxious. Right. In the case of the cell, the exhausts are free radicals. Correct. Yeah. Right? Oxidative stress and free radicals. And so the free radicals themselves will damage the mitochondria, will damage the cell, and so on. So we have all these systems in our body, like glutathione and peroxidase and all that stuff, to reduce the free radical damage. Antioxidants, right? That's why they are so powerful and important. And where do we get most of our antioxidants? From plant-based material, right? From fruits, vegetables, colored things, the carotenoids and all that. So that's why we need them, because phytonutrients are dying. Yeah, our cells are choking every single minute. I know. I like I said, I'm struggling with what's been going on in the dieting industry. I know that the pendulum is going to have to swing back. I know everyone's going away, went from vegan, now they're going carnivore. And then I am concerned about colon cancer risks with that as well. I mean, that proteolytic fermentation, when I studied that, I realized how detrimental that, and I see it on the Biome FX stool test Yep.
so many people are favoring that proteolytic fermentation.
Yeah, because we went to this phase where prior to the start of the carnivore kind of movement, it was like high protein, high protein, high protein, right?
Mhm.
People were like,
Atkins
exactly. They're like, and so
and fat.
yeah, between ordering a hamburger and just not eating the bun, just eat the hamburger. It doesn't matter what the quality of the meat is. People are going to fast food restaurants and ordering, Big Macs and just removing the bun as if now it's healthy in some way. And then they were just doing protein supplementations, right?
Right.
50, 60, 100 grams of whey protein every single day. And that became kind of the standard for a healthy diet. And then now it's kind of moved into this carnivore space. And what is dangerous to me about it is the way some of these diets come about is one person does it and it seems to have some benefit for them, and then they write a book about it, and then it becomes gospel,
Mainstream.
right? Exactly. It's like, oh my God, that one person wrote a book about the carnivore diet. They did it for 40 days and they lost weight and all that. I guarantee you, if you do keto, if you do carnivore and all, you will probably lose weight in the first 30 days or so. And the reason for that is not because of cutting out, plant-based materials or fruits and so on, is because you're removing most sugar sources.
Yeah.
Right. If you cut out sugar from your diet completely, however you do it, right, you will lose weight. That's inevitable. But your microbiome will start getting really significantly diminished over time, and long term, even 6 months long term, you will suffer from much higher risk of other conditions because your microbiome is being dysfunctional, is being decimated, because the vast majority of that fermentive base I mentioned earlier requires plant-based nutrients.
Yeah.
That's what
Yeah.
they evolved on.
Well, the fat can also promote endotoxemia.
Absolutely, right. So that's another thing. Like, fat was a luxury throughout the course of human evolution, right? There's a reason why fat smells so good to us, right? There's a reason why bacon.
I was just thinking about that.
Yeah, most people have a genetic affinity to wanting the bacon smell, right? The reason for that is because everything that is important to our species propagation, that is hard to find and takes work, our genetics created desires for those things to motivate us to go get it. Sex. Why does sex feel good, right?
Yeah.
Sex feels good because it's a lot of work, and it takes work to find the two people that want to do it together and so on. And so we are designed to be motivated to do that, right? Can you abuse that? Absolutely, right? There's 1,001 ways to abuse the desires around sex, as people do all the time. No different than why fat feels and smells really good, because just think about in the wilderness, if you were a hunter-gatherer type of person, how hard it is to go out and find fat, because fat you have to kill, you have to chase down, you have to trap, you have to kill. It takes a lot more energy. It's a lot more risky than just gathering and picking things and digging for roots and tubers, which is what they did most of the time. But your body does encourage you to go out there and find it from time to time, right? But it doesn't mean that you should be on a fat only diet or predominantly fat diet for any period of time, right?
Oh yeah.
Yeah, it's, and people bring up the Inuit Indians, for example, all the time, and I go, they're a very unique species of human that have not only evolved their microbiomes to be able to survive on a diet predominantly of seal blubber, but as it turns out, recent evidence suggests that they also have certain genetic SNPs that have selected for them among their population that can do better with metabolizing fat than others, right? Who get less endotoxemia and so on, right? And so, balance is the key. And you never want to say fat's bad, protein's bad, plants are bad. There's lots of movements out there that basically are villainizing plants, right? They're basically saying, oh, plants have these anti-nutrients in it and lectins and all these things, and so you can't eat any of these things. All the nightshades are bad, right? I think a lot of it can be detrimental to your system if your microbiome is unhealthy.
For sure.
And that's what it comes down.
That's what it boils down to.
Exactly. If your
ultimately
is unhealthy, almost anything that is even good for you can be a problem.
Tell us what you think about glyphosate. I know you've talked about it with that metabolic pathway, the bacteria that get hijacked.
Yeah.
And could that be one of the main reasons why we're seeing such dysbiosis amongst children even?
Yeah, I would say there's probably three big drivers of dysbiosis among children. Number one is the ubiquitous exposure to glyphosate, right? It's everywhere. It's in so many foods that kids eat. It's in the environment. It's in the water, and so on, right? So, and the big problem with glyphosate is it is an antibiotic. So Monsanto had actually filed a patent on using glyphosate as an antibiotic back in the late 70s. So they
Oh,
confirmed it is an antibiotic, but it's in fact more egregious than most antibiotics because it selectively kills good bacteria, right? That's what's so crazy about glyphosate as an antibiotic is that in your gut, a lot of the opportunistic pathogens like Klebsiella, for example, are resistant to the impact that glyphosate has, right? And then there's all of these beneficial bacteria, a lot of the lactic acid bacteria and so on, that are very susceptible to it. So if you're continuously exposed to this antibiotic through your food and your water and so on, over a relatively short amount of time, it creates a very significant dysbiosis. We published a study. We did a study with King's College in London, and this was the first study to show what happens to a pediatric microbiome when it's exposed to glyphosate. And we use food level of glyphosate, right? So this is the level that is designated by the EPA as being safe. And so in just 3 weeks of exposure to glyphosate, the pristine 3-year-old's microbiome started to look like a microbiome of somebody that had IBD, inflammatory bowel
oh
disease,
no.
right? Reduce inflammation, reduce diversity very significantly, significant reduction in short chain fatty acid production, huge increase in ammonia production, huge dysfunctional bacteria increase like Klebsiella and so on, and then started producing things like branch chain fatty acids that are more problematic and inflammatory. So it's completely changed the microbiome around in as little as 3 weeks.
3 weeks.
Right? So think about a child that's born and for the next X number of years, they are exposed continuously to this type of compound in the food source, in the water source, and so on. So that's one big driver. The second one is the overuse of antibiotics, right? Even the CDC estimates that at least 50% of antibiotic prescriptions are unnecessary.
Yeah.
And I've talked to lots of urgent care doctors about this because at one point, we were trying to work on a product that helps urgent care doctors or pediatricians because, as a father, I know that there's nothing worse than your kids feeling sick, right? You want to make them better. It's hard for you to see them suffering with fever and body aches and all that stuff. So what tends to happen is 90% of the time, these are viral things, and you go to the pediatrician and the parents put pressure on the doctor to give them a prescription, an antibiotic prescription, because they want a pill, right? They feel like if they're giving their kid a medicine or a pill that they're going to get better and they're doing their job. And so doctors are actually under pressure to write antibiotic prescriptions. And they haven't understood or been trained that antibiotics aren't safe. They have a side effect, right, of disrupting the gut microbiome. So I think the statistic is on average in America, by the age of 2, most kids have had at least 4 rounds of antibiotics, right? So it's incredibly disruptive to the gut microbiome.
Yeah.
So that's the second. So glyphosate, antibiotics. The third is the fact that around 33% of births are C-section, right? Some of them, they
That
have
was
to be,
me.
right? Yeah.
My 2 kids too. I have to be honest with you. They were born C-section. I wish I knew the things I knew then. Now,
Yeah,
I wish I knew it then, but yeah.
exactly. And we just don't know, right? People don't know. So, obstetricians don't know this stuff, right? They don't understand the impact of it. And then not only are kids C-section, but moms aren't encouraged,
The breastfeed.
educated on breastfeeding enough,
Right.
right? Like I know I have friends who are my age, so I'm 46. I have friends who are my age who were discouraged from actually breastfeeding and were told that that formula was actually better because it was fortified with things like DHA, right? And so they would go to great extents like wrapping down the boobs so it wouldn't produce milk as much, right? And it's more convenient to bottle feed, of course, and it's less
Yeah.
convenient. So, modern society,
Isn't the intestinal gut leaky? Isn't the intestinal gut permeable? And then you put all those dairy molecules through that, and isn't that problematic like that too?
you're born with your gut being leaky.
Yeah.
Your microbiome is not established yet. You don't establish the mucosal layer well yet, the tight junctions and
Same
so
with the blood-brain barrier too.
exactly, yeah. And so the most important thing for the infant is mother's milk, which not only contains, the nutrients an infant needs, but it contains up to 200 different prebiotics to feed and seed the gut microbiome. And it contains lots of microbes as well. So mom is actually a big source of, of fermented milk, if you will,
Yeah.
with prebiotics in it, a symbiotic, a true symbiotic.
Yeah.
And so those are the three big things. There's other things as well. There's
Of
of course
course.
chlorine and fluoride in our
Of
drinking water
course.
and all that stuff.
Bromines in our food, chlorine, fluoride, food coloring, we're set up to fail.
Exactly.
I have one last thing because I know those listening want to know about the bladder and they want to know about the vagina. And you're like, you guys keep talking about the gut, but you haven't gotten into this. So let's just finish this conversation about that. And the reason why we haven't is because, as you can see, the gut is 99% of the problem.
Mm-hmm.
Correct?
Yeah, that's exactly right. Yeah, because think about the bladder, the vaginal canal are all made up of mucosal tissue, right? So the lining of the bladder, the lining of the vaginal canal are all mucosal tissue. They're connected to your central, circulatory system. The immune system functions in those areas as well, and the immune system and the mucosal system are all centralized in the gut, right? So all the direction and the intelligence and the functionality on how the rest of the mucosal tissue in the body function all come from the gut, and the rest of the immune system functions comes from the gut as well, right? So if your gut is dysfunctional, it drives a dysbiosis in other mucosal tissues, your vaginal canal, your bladder, your lungs, your sinuses, all of those, right? So like chronic sinusitis, chronic upper respiratory tract illness, all of those things
Asthma.
tied to a dysfunctional gut, asthma, right? All of those create hyperactive airways, hyperreactivity among the immune system, and then it allows for dysfunctional microbes to exist in those areas versus the immune system being trained to know what should exist there and what should not. For women, it's a double-edged sword, right? Because the vaginal canal is so close to the anus, and so if out of fecal matter you're kicking out a lot of opportunistic and dysfunctional bacteria, you're going to get exposure of those bacteria to the vaginal canal, right? Yeah. So there's constant source of dysbiosis in the vaginal canal as well. And anything that you do in the body that compromises the gut can also compromise the vaginal microbiota. Antibiotic use, glyphosate, right? Sitting in tubs with chlorine water.
That's why when you take an antibiotic, you can get a vaginal yeast infection.
Absolutely, right? Because it gets through all the mucosal tissues, it's in circulation. Anytime your gut is dysfunctional and you're not producing the right compounds, the lack of those compounds can impact those ecosystems in the bladder, in the vaginal canal. And of course, the bladder and vaginal canal are connected, where the vaginal microbiota dictates in part what lives in the bladder, right?
Yeah.
That's the upstream effect of what happens in the vaginal canal. And then what happens in the vaginal canal is largely dictated by what's happening in the gut. So it becomes all connected. And there are ways of isolating and treating the vaginal canal and the bladder and so on, but you have to know that a root cause driver is in the gut, and it's counterintuitive, right? You're like, oh my God, I've been suffering from yeast infections, UTIs, bacterial vaginosis all this time. The last place you would think that you need to pay attention to is your gut, but it's absolutely connected. And in fact, there's some early studies that show that metabolic byproducts, for example, of estrogen in your gut actually go through the gut mucosa, can make their way into the vaginal mucosa and actually help seed the right types of organisms in the vaginal mucosa, right? So there's so many connections in there. We're still trying to learn, because we know that microbes in different parts of the body communicate with each other. There's something called the gut-lung axis, right? Because there's evidence now that shows that microbes in the gut influence function of microbes in the lungs. through a number of different mechanisms. And so microbes everywhere in the body will influence one another. And so you have to think about the largest reservoir of microbes, which is the gut. So.
Yeah, that's really good for everyone to hear that, because I think sometimes they try to isolate the bladder from the gut when they're evaluating. And I mean, that's why, I mean, I don't really like the PCR urine tests. People bring them to me constantly, and I'm like, and then they get treated based off the bacteria.
Yep.
And when people come and they're getting treated with antibiotics based off of what a PCR is telling them, I really have to question that. And people still, you know, they come to like a functional practitioner and they want this holistic approach, but yet they want you to treat like conventional. And I'm like, you gotta put that away, put that aside, pretend you didn't see that. Let's work on, build that foundation.
Yep, absolutely. The foundations are the key, right? And because that whole mindset of, oh, we tested it, oh, this is what the test says, it shows this pathogen or this microbe there, and then let's go after the microbe, as if there's a way of just going after that one microbe, right? We know that anything you do to go after microbes, you're going after all the microbes.
Yes.
Right?
Many of them,
which is,
that's why I question that approach.
Exactly. Whether it's a prescription method or it's a natural method, right? Whether you're using antimicrobials or you're using antibiotics, they're all going to go after good and bad microbes. None of them are selective for just bad microbes. So you know, that mentality of, oh, here's the presence of a bad microbe, let's go after it. It's flawed in the sense that you cannot just go after one microbe, right? It doesn't work. So you have to be prepared to deal with the fact that in your pursuit of going after that one microbe that you believe is there and believe is infectious, it may not be, right? Like you mentioned, there are imperfections with the way PCR and things are done, right? Yeah. In that pursuit, you have to be ready with the notion and accept it that you're going to damage more stuff.
Exactly.
You know, because we're dealing with an ecosystem here.
Correct.
And anytime you go in with something that's a broad spectrum destroyer of microbes, you're gonna dismantle the ecosystem to a certain degree, right? So and maybe you'll get the pathogen and maybe you'll feel better, and there may be a symptom relief. We see that all the time in IBS, you see that all the time in SIBO, you see it all the time in conditions in where they do a strong antibiotics like a rifaximin, for example, right, for IBS or SIBO. There's relief for a certain period of time, maybe 2 weeks, maybe 3 weeks, then it all comes rushing back, right? Again, because we're not going after the root cause problem. We're just, you know, taking that kind of allopathic approach.
Allopathic approach. Yeah. Well, it was a pleasure having you today. Thank you. I know we covered a lot.
We did.
Yes, we did. I wasn't expecting us to cover so much, but I'm really glad that we did, because it's all super important, and I've learned a lot today. So I really, really appreciate your time.
Really fun conversation. I'm glad we finally got to do this. We haven't done this before, right?
No, first time.
Done an interview before. We've had lots of conversations, but we've never recorded it. But it was really fun. Thanks for the opportunity to share this information. I think all of this empowers people, and it's super exciting that people can start to take their health in their own hands to a certain degree. Right?
That's what we want. We want to empower them. Thank you so much.
Thank you.
Well, that's all I have for you today. If you have any questions or specific thoughts, or think your gut health might be affecting your overall health, I'd love to hear from you. Be sure to like this video, share it with your friends and family, or leave a comment below if you have any specific questions. Be sure to subscribe to my channel so you don't miss any updates from me. Now, you can find the link below for my one-to-one root cause investigation program. And there's also a link below if you want to book a discovery call with me to see if I can help you optimize your health and reverse any health challenges you're dealing with. That's it for now. Until next time, bye-bye.